So, just a little bit of an outline. So, start with a little bit of background on Oplopanax, traditional uses, contemporary research, a little bit about macrophage activation, experimental design, results, conclusions, and then just some important issues, and all within the time, hopefully. So, Oplopanax horridus, it's also known as Devil's Club. It's a deciduous shrubby member of the Araliaceae family, or ginseng family. It also has names of Pacific ginseng, Alaskan ginseng, and wild armored Alaskan ginseng. We could see why armored. So lots of spines. It's considered to be one of the most important medicinal and spiritual plants, but by local indigenous people within its range. It grows in wet, moist, well-draining soil along streams and creeks and rivers. And it expands clonally through layering upright, woody stems that actually lay down and become decumbent, where new stems come out of those decumbent stems. So here we can see that this is Oplopanax, and it can get up to 20 feet tall. And the leaves are a kind of a maple leaf-like shape, and they can become 14 inches across. And here you can see, you have to kind of envision it because it is under the soil here, but there is a decumbent stem that's linking all of these stems of the Oplopanax. This is its range, so from Alaska, Canada, here, and then into the northwest part of the United States, so Washington, Oregon, Idaho, Montana, and also into Michigan. And in Michigan, it's endangered. Oops. So traditional uses, and these traditional uses are taken from Nancy Turner's paper in the Journal of Ethnobiology, which was in 1982, where she talked about traditional uses of Devil's Club. So its traditional uses include external and internal infections, including tuberculosis, arthritis, respiratory diseases. It's also been used to treat pain and broken bones, fever, cancer, diabetes, and most common preparations are decoctions or infusions from the inner bark of the stem. So contemporary research, OH extract has demonstrated antibacterial, antifungal, antiviral, anti-TB activities through screening assays that were performed by McCutcheon et al. There's also been some anti-diabetic activities that have been reported in animal studies, and anti-cancer activities, which have been shown in cell culture lines that are cancer cells, showing that it inhibits their growth. And antioxidant activities, which have also been done in cell culture lines. So macrophage, like the name, they're big eaters. So they're an immune cell in the innate immune system. They're an antigen-presenting cell, which means they eat bacteria and other particulates, and they actually present them to other immune cells in the immune system, kind of telling them, oh, hey, look what I have, go get this. So if you were to get an infection, the macrophage would eat that bacteria, and then it would present it to other cells in your immune system, telling them to go get it and kill it. So it's involved in many immune responses, including host defense, so prevention of infections, wound healing, chronic inflammatory diseases, autoimmune diseases, including diabetes. And macrophage produce chemicals called cytokines and chemokines, and a number of other chemicals or biochemicals that are involved in the production of fevers and activate other parts of the immune system. So this is the model for the macrophage activation. So in this model, we're using a macrophage cell line, which is called RAW264.7, which is an interesting name. But needless to say, then we apply lipopolysaccharide, which is a component of the outer membrane of the bacteria E. coli. And this lipopolysaccharide binds into a receptor on the macrophage cell membrane that's called toll-like receptor 4. When LPS binds to toll-like receptor 4, it activates a number of complex signaling pathways that then the macrophage will produce oxidative species, so reactive oxidative species, nitric oxide, TNF-alpha, which is another cytokine, interleukin-1, alpha and beta, interleukin-6, IL-10, and PGE2, and a whole host of other cytokines and chemokines. All of these are involved in chronic autoimmune diseases, diabetes, fighting infections, so with tuberculosis, wound healing, and a number of other physical processes. So the idea was to collect Oplopanax, horridus, bark, and then test it in the cell model to see what happens with the LPS-stimulated macrophage cells. So we harvested the Oplopanax horridus from the Skagit River watershed, and this is just a little map here where this is Washington State, and then this is where the Skagit River watershed is. And it's a pretty healthy watershed. Skagit River has all 5 species of salmon running it. The inner bark was then peeled off of the woody stems and then ground into a powder. And then the Oplopanax powder was then extracted in ethanol and then sterile filtered and resuspended in a cell culture media. So let's go back, sorry. So here is just the plant that it was harvested from. And then this is the bark being peeled off the woody stem. And then these are the pieces of the bark that were then ground into a powder. So these are the RAW264.7 macrophage, which were grown up in a lab, and then were stimulated with LPS or Oplopanax extract in varying amounts, or both at the same time. And then they were incubated, so they were put back into the incubator for 24 hours. And then at 24 hours, they were taken out, and the supernatant was collected. So the cells, the macrophage cells, are actually not collected in this experiment. It's just the supernatant, and this contains all the cytokines and chemokines that we were measuring. So we harvested the supernatant, and then we did the cytokine chemokine analysis and the PGE2 analysis, and then we analyzed that data using GraphPad software, where we analyzed it and graphed the data. So in this first figure here, this is our HPLC chromatogram, so showing polyenes and polyines that we're not for sure that is exactly what they are. However, it is consistent with the previous HPLC chromatograms that have been published. So here we have the graphs. So TNF-alpha production, IL-1 alpha production, IL-1 beta, these are all important cytokines in the fact that they also produce fevers. So here we can see this here is our positive control. So all the red here are positive controls. These graphs are a little confusing, but these are all controls here. And then here is where we have the LPS plus the Oplopanax extract. So we can see that with the TNF-alpha production, we start seeing that it is becoming inhibited at 250 micrograms per mil of Oplopanax extract. And then here with IL-1 alpha, we're starting to see a significant inhibition at 12.5 micrograms of Oplopanax horridus. And same here with interleukin-1 beta, we're also seeing it at 12.5, and it's significant inhibition. And these are both, we can see here, that these are dose-dependent, decreasing here by the graph. And here we see with IL-6, we're seeing a inhibition at 12.5 micrograms per mil, and our p-value is significant at 0.001. And then IL-10 production was also inhibited at 6.25 here, and that was significant. So we also find that, that the prostaglandin E2 production from the macrophage, PGE2 is an inflammatory mediator, and it is produced by COX enzyme, so COX-1 and COX-2. And aspirin is one of the known inhibitors, along with ibuprofen. So we can see here that the LPS is the positive control again, and then we're seeing a significant inhibition just at 6.25 micrograms per mil. So that's pretty significant, and it's completely gone at the 25 micrograms per mil. Okay, so conclusion is that Oplopanax horridus extract inhibits TNF-alpha, IL-1 alpha and beta, IL-6, IL-10, and PGE2 in an LPS-treated RAW264.7 macrophage cell line, which is a model that is consistently used to test medicinal plants and their effects on the immune system. So a lot of drugs that eventually are getting studied start here in this model. These activities are consistent with its traditional use, and more research needs to be performed to fully elucidate the mechanism of action of these immunomodulatory effects because of the complex signaling that we saw. We don't know exactly what's being inhibited, and also to identify the biochemical constituents within Oplopanax horridus extract that's responsible for these effects. And further studies will include anti-diabetic and anti-obesogenic assays. Okay, so important issues. In 1997, this is the last date that I can get an estimate, 2,000 kilograms of bark were harvested from British Columbia wilderness. This bark is used, obviously we're not completely sure, but a lot of it can be sold to supplement industry, and it may be being collected for personal use also. So because of the way that Devil's Club grows, ecologically sustainable harvesting techniques are required, otherwise you will kill the plant. And traditional intellectual property rights are also vitally important when looking at the commercialization of Oplopanax. And to ensure that Devil's Club is used in a culturally appropriate manner and that intellectual property rights are protected, it is vital that First Peoples control the growth and direction of any further commercialization of this plant. And this was taken by T.C. Lance from an occasional paper out of British Columbia. And that's it. So I have some acknowledgements. I have some collaborators at the University of Montreal, and so Leigh Joseph, and then a number of other people that we're working with, and David Kiefer for helping peel the Oplopanax, and the Bastyr Research Department, and also Bastyr Research Department faculty seed money grants, Dr. Turner, and Indigenous populations that shared their knowledge with Dr. Turner. I
think we have time for one question.
Yes?
Yeah, one thing I'm interested in what you're talking about here is that Devil's Club is a species that's pretty well known to have been widely distributed by the tribe, that everywhere that they traded it and everywhere they went they planted it, because its distribution does not conform to any standard ecological approaches. So
right.
I'm wondering if why they're trying to control this intellectual property when traditionally it was used very widespread and used by all the tribes and for similar sorts of purposes, and they were planting it where they went. It didn't distribute itself naturally. Well, if you consider humans as a non-natural distribution form, but they were distributing it to make sure that it occurred all the places where they thought they would want it or need it.
Right. So I think it's mostly because it is such a significant plant for medicinal and spiritual use. that that definitely having the indigenous tribes that have been involved in its in the intellectual property rights is important to have them involved.
Sorry, it's okay. Thank you so much.
Are not interested in other people making money off of it.